It's a really cool kind of fish. I think they're really cute.
And this is scales on head or scales on not on head? What's the
Push in.
Yeah, so if there are no scales on the nose, then it's Aldrovandia. If it has scales on the nose, then it's Halosaurus.
All right, let's get
So
a
this
shot
will,
at the nose. Oh.
yep. There were no scales. Yeah, it looked really see-through.
Startled it.
Yeah, they're really fast. I think we got a black coral.
What's up?
Hi. This is Stauropathes.
Coming up.
Yep, got a hermit crab. He's running so fast. So this is a Parapagurus, a type of hermit crab that has a very special shell, as you see. Kind of looks like a glove. It's wearing a zoanthid, a coral, as a little backpack, so it doesn't actually have a shell. It uses this zoanthid as its shell, and it holds on to it, and they're buddies for life. Isn't that cute? So they grow together. So this crab will never have to find a new shell like other hermit crabs that have to size up.
Dan, are you there?
Does this zoanthid only grow on this crab?
I only ever see it on this crab, right? Yeah. I'm not sure
Dan,
of
are
the
you there?
species of the zoanthid, but we usually always see them together like this.
It's really cute.
We learned on a different cruise when Trevor tried to suction this as a sample that the crab can let go of its backpack if it wants. Yeah,
It can?
it totally
so
can.
Unfortunate.
Yeah.
Would it go find, I mean, I guess you don't really know, do you, if it would go find a new one?
I don't know. I don't know how they find them in the first place.
Yeah.
All right, come on.
Yep.
But like any hermit crab, I'm sure it could go and find a shell.
Yeah.
It's just that these parapagurids, they almost always have a cnidarian as their shell. So either an anemone or a zoanthid.
Little buddy.
Where am I going?
A viewer wants to know how the crab and other deep sea animals reproduce.
So some animals are brooders and some animals are spawners, but the short of it is they eject their larvae into the water column. So some animals will release their gametes into the water and they'll mix in the water column, and some will meet up and do it a more traditional land way. But in any case, most of these animals produce larvae that will float through the water and then settle out and recruit later. I think that was a different Psychropodes, that purple cucumber we went over had the shorter little tail.
Don't really want to mess with cycling them if they're
Yeah, I
down
think
there.
they're fiddling down there.
Yeah.
So
Yeah, a couple notes to add to that question on what Megan just alluded to. So for those species who are spawners, so releasing both the eggs and the sperm into the water column, it's something that's an active research, area of research, reproduction obviously being one of the most fundamental processes for the survival of a species long term. As you can see here, the densities of animals are much, much lower than what you would see on terrestrial ecosystems or shallow water ecosystems. So how does that work when you have so much fewer animals and you're releasing eggs and sperm into the water column? It has to be timed exactly at the right time, and how do these things find each other? And there's things that we know very little about. Obviously, the animals are successful at doing it, but it's also very hard to study this in the deep sea where there's very few observations.
Push in on the sponge.
So this is a euplectellid sponge, a glass sponge in the family Euplectellidae. It is in the subfamily Corbitellinae. And there's a little polychaete on it, I believe. You see a little pink spot.
Oh yeah.
I can tell it's this this type of sponge because of the texture of it. It kind of has like this loamy texture and very crisscrossed looking spicules as we look down into the sponge.
Oh, flying,
Whoa, what was that?
flying crab.
Little crab.
All right,
Cool, thank you.
come on. And they're back. Not choppy.
Yeah, just a little bit for context here. So we have sponges. It's a large group of animals. They're currently in estimate over 10,000 species. They are the simplest of the multicellular animals that we have. And yeah, they also have a few interesting, a lot of them are filter feeders, taking out of filtering the water and getting out plankton and tiny. They also have cells that are totipotent, I believe is the Latin word. They can, any one cell can become a different type of cell. But there's also a group that is very poorly studied for the amount of biodiversity there is. So a lot of corals and sponges and other invertebrates get a lot of the attention, and sponges is a group that's poorly studied, and they're also difficult to study because they also have high plasticity, meaning that the same organisms can look very different in a different environment.
Yep, another crab.
Yeah, let's get
Did
a
you
zoom
see
on
it
that.
swimming?
Yeah.
That's cool.
Can push in there. Oh, it's fast.
This one's really speedy.
I remember trying to catch one the last time I was out here. It was
They're
quite
hard to
an
catch.
event. We successfully caught it though, and it was like finally when we did, it was everyone like erupted, like
Yeah.
the game-winning point scored at the end.
So a question on this crab. What do they eat?
They eat like small crustaceans like amphipods and isopods and polychaete worms and pretty much anything they can get their little claws on.
Stirred up some sediment.
Ooh, that was really cute.
Oh no.
It's like, hide in my shell.
Oh, that's cool to see the underneath.
All right, we can let this one go.
Okay. Come on.
little update for folks that might have joined us yesterday. We're pretty early in a dive. We came across a large gun casing, a shell casing of ammunition early in a dive, and we had several of our archaeology scientists ashore chime in and send various emails and texts overnight. And the consensus seems to be that this is was a 40 millimeter casing, most likely from an MK1 Bofors round. We used an anti-aircraft artillery during World War II.
Fish.
Oh, that's a chonky fish. Big one. So this is an eel in the family Synaphobranchidae. And as we get close, we're going to look at the dorsal fin and the anal fin and how they look in relation to each other.
Push in.
And that's going to tell us a little bit more about the genus and species of this animal. So when the dorsal fin starts before the anal fin, it's an Iliophis. And that's what I think we have here. You see how far forward that dorsal fin starts. It also has a very skinny nose and very pronounced pores on the lateral line. And very small pectoral fins. So this is an Iliophis, or arrow-tooth eel.
Do we have an idea about the size of the eel?
So those green lasers are 10 centimeters apart. So this one is probably about half a meter, a little longer.
Yep. I gotta keep going.
Yep.
Moving up the slope.
Yeah, some of these eels can get to be about a meter in length. Synaphobranchus riverdorsalis, species that we often see a bit deeper. We see them at this stuff too. They tend to be the biggest of the cutthroat eels that we see. And they're very distinctive because their dorsal fin starts way, way back on the body. So it looks like almost like they don't have a dorsal fin. It starts so far back.
Yeah, and that group of the cutthroat eels, the common names comes from the location of the gill slits, who are right,
There's a umbellula.
Right on their throat, giving the common name of the cutthroat eels. But yeah, this is a primarily deep water group
It's right next to the rock.
of eels
Yeah.
with a wide geographical distribution, and I believe the biggest ones of these have over a meter and a half.
Push in there.
Umbellula.
Yeah, this is a sea pen. Oh, and it has a little, oh, okay, there you go. So beautiful. You can really see the pinnate tentacles. So these are octocorals, meaning that they have 8 tentacles. That's great, Jake.
Okay.
Thanks.
Maybe a fun fact for maybe listeners at home. So the term corals that we use is it's really a term that we use to describe a whole bunch of organisms that have a similar shape and that are somewhat closely related, but really underlined somewhat. So there's many, many very distantly related groups of animals that we use the term corals for. So they include the stylastrids, black corals, Gorgonian corals, so multiple different classes of animals. So it's It's really a term more related to the functional morphology of these animals. But they are all cnidarians, so animals that have stinging cells and that have either a polyp and or a medusa life form, and some of them you have both, and some of them you have one of them more pronounced than the other.
Halosaur?
Yep, another halosaur. It's another Eldrovandia.
Quick snap zoom? Maybe not.
Yeah, they're tricky. I see how cute. So cute. Boop. It's a stick. There's so much life on this.
Is that an urchin?
Yeah, there's a few urchins on here. There is a small sea star. There are munidopsid crabs. There are serpulid worms. There are anemones. I think there's a snail. There's a scale worm. There's an octocoral. There are a bunch of brittle stars around this area too. This is a lot of food, so it's attracted a lot of animals.
Is this the same urchin we had a warning about when they collected the sample yesterday?
Oh, I don't know.
Was the question, is this the same urchin that we collected?
Yeah.
No, the urchin we collected yesterday had very long spicules and a much smaller body. It was kind of a purple-bodied urchin.
Like an
Yeah.
Elpidiidae.
Could be.
They have like super long spines.
Yeah, sort of thin, long long spines. These ones are quite small and compact. So definitely different species. That was the escape urchin that kept trying to come out of the box.
Yeah, they can kind of take off because they're very neutrally buoyant. We've been seeing a lot of these Aldrovandia, or is it the same one? It just keeps getting ahead of us. Oh, look how pretty those scales are. Such a good color.
This one looks really peaceful.
There, you can definitely tell there are no scales on the head.
Oh.
And if I remember correctly, there's this is a pretty wide-ranging genus. I want to say they're found in all oceans, about a half a dozen or so currently described species, and all with a pretty wide geographic distribution.
A viewer is asking, how far are we from land for a stick to appear at these depths?
So yeah, we're on a underwater mountain, so you'd have to go first 2.5 kilometers up, and then you'd have to go about 60 nautical miles to the northwest, and then you would come into the main island complex, so that's the closest point of land here. But yeah, wood obviously can float and can drift long distances, so it's not uncommon to see wood at these places where you're far removed from the source. And as Megan alluded to, these wood falls are important food sources. They can persist on the seafloor for a very long time and provide food that is a lot more abundant than what you typically have in the background state in the deep sea. And it's interesting also, just ecological phenomenon. So wood falls and whale falls also, or bigger fish, when they fall, they provide a rich source of nutrients. And there's actually species that are specialists to just, that's how they get their food, is to get live on these things. And so yeah, that begs the question: how do they find these sources of nutrition which are not there? And I should say it's not a lot of these are also just general feeders. They'll scavengers. They'll eat what they can. But nevertheless, a lot of deep sea species have a very acute sense of smell and very good at finding the few sources of food that are found here in the deep sea.
Yeah, and debris from land like sticks and coconuts and other objects are known to travel quite far with tropical storms. So even when we're really far offshore, sometimes we see natural debris from terrestrial settings and other sorts of debris as well.
A follow-up on that, Do we know how long it takes for the wood to absorb water and sink?
I think it would depend on the wood, the size of the wood. Like if it were a small stick, a large log, and yeah, what kind of species that tree or plant used to be. But they some could float for quite a while, and then sink quite offshore.
Yeah, and it's just interesting for anyone that has ever come across a piece of wood at the surface in an open ocean environment that's been there for a while. They'll start aggregating all kinds of things at the surface as well, and then you'll have a lot of fish that will, hover right underneath it. And so yeah, they'll aggregate things on them. And as you can see, the same things happens then once they reach the seafloor. A lot of things will start feasting upon the nutrients they extract from the wood.
Oh, Hypnops. There's one off to the right there. See the little straight line with the white. So this is a really cool lizard fish called Hypnops. It doesn't have true eyes. It has these sort of vision plates where it can just detect light. So those are those yellow, white scales on the head. They're ambush predators, so they'll sit and wait. They're related to the tripod fish.
Yeah, and if I remember correctly, there's a theory that those structures there, by the eyes, that they actually emit. They have organs to emit light, and they use that to attract prey. And several years ago, we did a test on this to basically turn off the lights of the ROV and see if we could pick up on the UV light that they emit. But yeah, we couldn't see anything with the cameras at the time. But yeah, I believe there's a couple papers out on that about the eyes not being able to detect but emit lights and use that as a lure, a light lure.
Oh, that's really cool.
What is the common name?
It's like a deep sea lizardfish.
Lizardfish.
That's great, thanks. Yeah, they're very distinctive. You have to look for this, like, it's so super small fish, but with those eye plates like that, they stand out. So I knew right away what it was. I believe we collected one yesterday.
Yeah, we have a lizardfish, an Hypnops.
And then you also have a Bathy Terroy. Or no, you had a, what was the other one? There was another lizardfish that we collected. The blind one.
A couple dives ago.
Yeah.
Yeah. Yesterday we also got a batfish.
Yeah, the batfish was cool.
Yeah.
I'm here for the really weird fishes.
Same.
Gotta catch them all.
Yeah.
or just a few? Just a few to
Just,
know.
we'll gotta catch them
Who's
all with the
there?
camera.
Yes, that's one way to put it.
We do have a viewer that says Ipnops should be a Pokemon name.
I'm surprised it's
Sounds
not
like-
a Pokemon
yeah.
name.
The first time I saw it written, I thought it was a misspelling, but it's a very cute name for a lizardfish.
Mm-hmm. Though I think my favorite fish name is Boops Boops.
That's adorable.
Yeah.
Boops, boops.
Yep.
Does it match its name?
They're pretty cute.
Take a quick look at that dark object there.
Though I know some viewers have suggested that what I find cute might be concerning. But that's okay. That's just who I am.
Looks like another piece of wood.
I feel like we've been seeing a lot more wood on these dives than I've seen in many of the dives that we've done previously in different seasons. But a lot of the exploration has been done in ultra remote islands, whereas here we're a lot closer to large mainland masses. So maybe that has something to do with why we see more woodfall here. But I don't see this like a lot, say, in Hawaii. Or it could just be the luck of the draw, you know? The deep sea is very patchy. And we've only explored a very, very small fraction of it. So what we see, we're lucky to see.
Yeah, and one thing on that, we are, of course, a lot closer to bigger land masses here. And the other factor, too, is that there's a really strong current. So at the, around Palau, you have both the north equatorial current coming from the east to the west, and then the north equatorial countercurrent going from east westward, so basically going straight from the Palauan Island complex coming out here, so kind of serving as a express highway of sorts, could bring things further offshore. And also one note I wanted to make here on the importance of these specimens collection. So a big part of this mission is to just do basic characterization and understand who is here. Understanding what's in your zoo is the first order of business when you manage a conservation area. There have been a few
Can I give you a DVL reset?
deep sea explorations in this area. Some of the more prominent ones were done by Jamstack here, and then most recently work by National Geographic Pristine Seas. Neither of which really included a large focus on collecting biological specimens, so big focus was there on geology and collecting video imagery, and these specimens, having physical specimens is really, for those folks who study taxonomy and naming species and classifying species, still the gold standard is if you wanted to describe a species, you need to have two things. You need to have a specimen, and that specimen then gets put in a museum, and then you need to have a description, so a paper with photographs, and that paper would compare that specimen to everything that's been described previously to ensure that it is
Ooh, that
indeed
one's
different
weird.
and unique. Yeah, can we get a
Zoom?
zoom in?
Yeah.
Sure. Push in there.
This one has fantastic
I dusted it.
texture.
Sorry.
Well, we'll wait for
I
it.
didn't think I was that close.
Daniel, do you think there's a future for taxonomy where a physical collection won't be needed?
Yeah, excellent question, and we are starting to see that. So there was a a recent paper description that was without a specimen. And so obviously now that we're getting much better imagery. Now, previously when the science was done, yesterday we were talking about the Challenger expedition, the first deep sea expedition that happened 250 years ago, long before there were photographs,
Yeah,
and
these
back
ones
then there
don't
was these
startle as
elaborate
easily.
hand drawings. Now we have high quality photographs, and we have ways to develop digital models of specimens, and we can collect environmental DNA, collect DNA of these organisms without collecting the organism itself. So it is certainly possible, and it's a movement, obviously, I think that is important, but we're not there yet. And these specimens do provide a lot of value for a lot of scientists. But that's an excellent question.
Do we want to look at it?
What did you think of that cucumber, Daniel?
Yeah, I mean, it's from the watches I've been, this looks different than anything we've.
Yeah, this is a
So,
new one.
yeah.
I haven't seen this one yet before.
Yeah, so I think
Get a look without
I'm
landing.
leaning towards, if this is if we're set up to do so, I
Collectible?
think a collectible. Yeah.
Okay.
Yeah, this is
Collection.
in the family Elpidiidae, so related to the sea pigs. It kind of reminds me of the one we collected, but this one has far fewer projections along its body, so definitely a different species. And you know, this has been like sea cucumber heaven. There have been so many different species of cucumber that we've been seeing, and a lot of them are very different from what we've seen in other parts of the Pacific.
Jeff chimed in and said we may have collected one on the first dive.
Did we
Maybe
get one
could-
of these?
Maybe we could get a close-up zoom.
Yeah, I just
first
wanted to get a good
before
look
we-
at it.
Push in there.
So great. What a cool highlight.
It's got really fantastic texture.
It does look quite similar, but
It could be in the same genus as the one we collected,
yeah.
but there are far fewer projections on it. We don't
Yeah.
necessarily need to collect it, but I don't want to pass it by if it's important.
I think, yeah, Jeff is also thinking it might be different, so I think if we're set up to do so,
Okay.
then let's go ahead and do so.
All right.
Yeah, there's also a scale worm underneath it.
Is this a slurp?
Oh, I think it's too big to fit in the slurp,
Okay.
but you might be able to use the slurp as a suction
Yeah.
to stick it in the bin.
Okay.
You can really see how soft the sediment is all around it too, which is prime sea cucumber feeding zone.
Probably forward.
Yeah, we are currently fully open. We only have one Niskin, so can put it in the forward if you'd like. Something nice and close.
And since we know this one doesn't like to do crunches, we can open the bin again sometime later.
So a viewer wants to know, how do we choose which specimens to collect on a dive?
Yeah, we'll just get here, check a second, and then we'll answer that question.
Do you want me to zoom? Or are you happy?
I think I'm happy. Yeah, ready. Okay, yeah, you can increase the PRV. Yeah. a little bit, and then let's try suction there and see what happens. Perfect.
That's so satisfying.
Yes, if you could.
Oh,
All right, pop out the box. All right, can stop suction. Can close the box.
Excellent collection.
and what was the sample number?
It's 65.
65.
Yeah, so thanks for that question there, Imade. So when we do these types of explorations, particularly in places like this where we're working in a very sensitive marine environment, and we want to make good decisions in terms of collecting, and so the samples that we collect, we try to prioritize those that have a significant discovery potential. So things that may lead to the description of new species or a significant range expansions of known species. So here in Palau, as I was mentioning before, so there have been very little, there's been a lot of, like in every tropical island system, a lot of research has focused on the coral reefs, dating back, many, many decades. But if you go into the deep sea, there's a lot less. And so we still don't know what's down here. And so one of the first things that we do is try to compare the images that we're seeing in real time to various species identification guides to see if it matches anything that might be described. And we have on board here various taxonomic experts and both generalists and specialists that know their groups very well. And then in addition to that, we have a small army of scientists ashore, which also are watching the live video feeds in real time and then helping us chime in with what they think that is that we're seeing, and if it is indeed something that might be new or undescribed. And so, yeah, we do not take these decisions lightly. We want to make sure that what we do here advances science, advances conservation, and advances marine management. And so we use the best available information to inform what specimens we collect. And then in addition to making the best possible decisions, we also ensure that the data samples that we collect on this mission is made available to scientists and from around the world.
Can we make a 30-meter move, 165?
So all these biological specimens end up with a partner that we've been working on for 11 years now, the Harvard University Museum of Comparative Zoology. They have a very wonderful archive of specimens that have been collected for centuries. They make sure that samples are taken care of and managed in perpetuity. They also have avenues for scientists from around the world to go request those samples. And if you request a sample, and they will send you that sample out on loan so you can study it in your own university or institution.
The shark of the cucumbers. Bono.
Daniel, do you have a sense, what are our latest you don't have to have this memorized, although I wouldn't be surprised if you do. What is the ballpark number of publications that have come from Nautilus expeditions?
This floating thing.
Floating thing. Oh.
Yeah.
Yeah, so the Ocean Exploration Trust and Exploration Vessel Nautilus just celebrated our
Push in there.
15th year anniversary. I've done expeditions.
Oh, it's in a Beluga,
Oh, wonderful.
but it's all closed.
Around the world, starting in the Black Sea, Mediterranean, and then working in the Atlantic, Caribbean, and then over the last 5 years in the Pacific. Mapped over 1 million square kilometers, over 1,000 successful
Right, that's
remotely
great, thank
operated
you.
vehicle
Okay,
dives, serving depths of close to 6,000 meters and to the surface. And I just actually tallied up yesterday peer-reviewed scientific publications that used data collected on EV Nautilus expeditions. We just yesterday surpassed important milestones, so 350 science publications and counting, a lot more in the way, and these cover all disciplines from the biological sciences, archaeology, chemistry, physics, sociology, and anthropology, and yeah, that's a big part of our model is that we try to make the data and samples available to the broader scientific community, so to help catalyze more research even after our ship leaves an area. And so on our website, there's links to all the data archives where people can download data sets. And yeah, that's really a big part of our model of how we try to do data is go to a place that hasn't been explored. Yeah, that's really interesting. Can we? Huh.
It's really strange.
That is.
It's just a
I thought it was a
crack.
stick at first. It's just a crack. Yeah.
Huh.
It's like a crevice or something.
Yeah, it looks like
Yeah,
a...
that. Yeah.
Maybe it was a burrow, and then the top of the burrow got scoured away.
Looks like an ice crevasse.
Yeah, if we can.
Is there a line through, like...
Yeah, the tightest zoom we can get. I mean, that's just very strange.
That's the tightest we can get.
Interesting.
Almost look like monofilament in there.
Yeah.
Yeah, that... It does.
I love these moments where we get stumped.
Yeah. Very puzzling. I mean, I see a shrimp in there.
Yeah.
It's a nice place to hide.
But what made this? It just seems too big to be. Yeah, I don't know. A little puzzled here.
Yeah,
Yep.
that's
Yeah.
pretty strange.
It's like almost 40, 50 centimeters or something like that.
Yeah. And on the bubble cam, you can get really nice.
Yeah.
Yeah, if we can just maybe just pan down and all the way up, maybe slowly see if we can get any more clues.
If we brighten up the image, it might like blow out the sand, like blow out the white of the sand, but maybe it'll bring a bit more light inside the crevasse. Do you think we could try that
Yeah, I can try that.
with the iris?
After they do the scan up, I'll try that.
Well, I think that filament is the
Yeah.
antenna of that shrimp.
Oh, it's just a shrimp and that
Yeah,
was-
there's a shrimp in there.
Okay. Maybe not then.
Huh.
Strange.
Tell that down.
I was checking here if any of our scientists ashore might have some clues of what we're looking at. Yeah, I mean,
Yeah.
it just seems too sharp too. I mean, the only thing I can think of is some kind of billfish, but yeah, that just doesn't seem right. Yeah, I think the only thing, if we can see, if we can play with the light a little bit, see if we can get a view inside and
There's definitely a shrimp in there.
Yeah, that's, I mean, that's about, I can go brighter, but
Yeah, I think that's plenty.
Well, we should probably get moving.
yeah, I think so too.
All right.
yeah.
Thanks, yeah, we got some good video and good close-ups. Also testing out our cinema camera here, got some good stills. Yeah, thanks for that. Before we came across that burrow, I was just a little bit about the importance of making data publicly accessible and
Oh, there's a star.
discoverable. That's a big part of.
Let's take a quick look at that.
All right. Going for a quick zoom.
This is Zoroaster, I believe. Thanks.
All right. Come on.
Yeah, just back to the importance of open access data. Deep sea research is obviously very logistically complex. You know, it requires advanced technologies, and it's costly. And you want to make sure that the data that we collect supports as much science and management and exploration activities. And so making that data available so that future scientists, students, educators and everyone, really that wants access to this data has that access is important. Making it easily discoverable and also interpreting some of these things is important, and that really also highlights a big part of our mission, the Ocean Exploration Trust, and also of our sponsors of the NOAA Office of Ocean Exploration and Research, is collecting this data and then also using the excitement of discovery and exploration to engage audiences of all ages, promote STEM-related fields. It's a big part of our journey and our mission.
Absolutely. Great time to plug if viewers are listening and are interested in joining our team. Our applications are open for 2025 to join the Science and Engineering Internship Program. If you're interested in being an ROV pilot, a video engineer, a data logger as an ocean science intern, or a seafloor mapping watchstander as a seafloor mapping and hydrography intern. These are paid opportunities to come and join Nautilus wherever the ship is in the world and train alongside our team. As well as our Science Communication Fellowship being open for educators, artists, journalists, community educators, folks that want to help bring deep sea stories out to larger communities. Erin, you started as an intern, is that right?
I did, yes, back in 2017. Can you hear me?
I can.
Okay. Back in 2017, I got the internship, and it's really hands-on. I got to film the Deep Staria, and then I just kept contracting from there. So I highly recommend it. And yeah, it was a great experience.
Jake was an intern as well, right?
I was. 2019.
I have to put May on the spot.
May.
I was also an intern in 2015.
And I was a Science Communication Fellow in 2013.
And on people not on watch, we got a few as well. Renato Kane, our mapping coordinator, and
He was an ocean science intern 2013.
Drag Atalanta a bit.
I was a high school
Come out of auto heading.
Nautilus live watcher
That's awesome.
before becoming an intern, so
That's so amazing. I absolutely love that.
Can push in there. It's gonna be a bit bouncy.
It's very fast. So this is a cusk eel.
Nice.
I think it's the Lucio brutula. Jeff will probably chime in.
What do cusk eels eat, Megan?
They eat small invertebrates. So things like polychaete worms and amphipods and isopods, copepods, like any small little invertebrates that I can catch, maybe other small fish.
Come on.
yeah, Jeff says it's a, oh, can we get a zoom on the head?
Yeah.
He thinks it's a Basio gigas or gigas,
Passagigas.
or a Lucio brutula.
So it definitely has sort of a scalloped head. That's what I noticed. This one's not being very cooperative.
No, it's not.
We're going to make you
Push in there.
famous on the internet. Let us see you.
That's not everyone's dream.
Fair.
Oh yeah, upper cam sees it quite well, but it's, we're also like way stretched out.
Someone away and I'm dragging Atalanta. All right, push in.
Look at those
That's
eyebrows.
fantastic imagery.
All right.
Yeah, the head looks kind of compressed. That was a challenging one.
Yeah,
Sometimes you
great
just gotta
headshot.
pull Atalanta along with you.
What did you land on as our most likely ID?
I think we got Basso gigas or Locio brotula.
Yeah.
Go back into auto heading and
refined with that zoom.
try to come around. It might not let you.
And for our
Jake?
Yeah?
We have a question for you.
Okay.
Did you have any prior experience with ROV piloting before becoming an intern?
I had none. No experience. I came out as an intern. Yeah, it was new experience and immediately was hooked.
What was your degree in?
Ocean engineering.
Oh, cool.
I think the Nautilus internship and science communication fellowship programs are really unique in that a lot of people who join them start off with no experience at sea, and it's one of the few programs I'm aware of, or maybe the only one that supports opportunities and pathways for individuals from many different backgrounds to get experience at sea.
Yeah, and then just want to take the opportunity, a big shout out on our current expedition. We have 3 incredibly passionate and talented interns in both our ROV video and our ocean science team. So Jaden, Max, and Ayla have just been doing a phenomenal job jumping right into it. And it's just really, really inspiring to see. Often when I see folks with that kind of enthusiasm and great spirit and taking active roles in their own future, it's just wonderful to see. So for those listeners that might be still thinking about their career paths and what they want to do in the future, and the one piece of advice is, follow your dreams, study hard in school, and then don't be passive in your own destiny. Take an active role in your own destiny. And these internships are a great opportunity. And as Megan alluded to, we currently have open applications. Those will close on January 3rd. And these are paid at-sea experiences for students and educators to come join us on one of our expeditions next year aboard EV Nautilus. It includes the time at sea. It also includes some training before these cruises. And so it's a great opportunity. And we have on our ship, in addition to the interns, we have our several alumni that have come out and become our regulars. So it's a great way to get involved.
Also, a paid internship in anything filming or video is very rare. So it's a really cool opportunity.
So 2 questions on the internship. One, do we need to have a degree to join or to apply? And then the second question, are there any openings on the sonar team for an intern to learn how to capture, process, and interpret like the multi-beam data?
Super good questions. Yeah, our internships, you do not have to have your degree done. You can be in progress, and in progress whether you're at a community college, in progress at a technical school, undergraduate, or you can apply if you're a graduate student as well. So really wide open, as well as early career, which we define as within the last 3 years of being in any one of those programs. So really wherever you are kind of getting your start, you don't have to wait till your degree is done. In fact, the vast majority of our interns are still students at the time. Because Nautilus's seasons are so long, you also can, in the application, indicate when you're available. So we totally know that people have a lot of commitments in their life, whether that's to school or family or community. So whether you are definitely available all summer or not available all summer and would rather be in a shoulder season or a different time of year, that's part of the application to indicate your availability. And yes, one of our tracks is seafloor mapping and hydrography. So you can pick which program to apply for, which really speaks to your interests and your strengths.
Yeah, and the cruise that we just had right before this, that was a lengthy mapping cruise that brought the ship all the way from American Samoa via Howland and Baker all the way to Palau. It covered an enormous area, over 4,000 miles of seafloor, and we had 3 wonderful interns, seafloor mapping interns aboard. And yeah, so as Megan alluded to, and that we have internship programs in various tracks, and it's also part of our staffing model here. On board, there's about 52 people or less at any given time. About a third of those are our crew. Those are the permanent engineers, officers, and folks that man the ship itself and the engines and cooks.
There's something to look at.
Yeah.
And the other 2/3 are our mission team that we rotate depending on what the mission is.
Pushing on this shrimp. Oh, never mind.
It hurt you.
And in every one of our, I'll just pause here so we get some good footage.
This is a penioid shrimp.
Now you can push it. Oh, never mind.
In the family Aristiidae. They are quite good swimmers, very fast.
Right.
They're scavengers here in the deep.
Was there also one of those long-legged shrimps?
Yeah.
Off to the,
Yeah, there was a nematocarcinus right there.
yeah, there it is.
Yeah, there it is.
Look how long those legs are.
One shrimp to the
Oh, it's
next.
swimming.
Too much dust.
They rarely swim, they usually walk, but
Are those called swimmerettes underneath them?
they're called pleopods.
Pleopods. Fabulous.
See, this one's not as good a swimmer.
Why so?
It's got all those legs to carry around.
Yeah, the leg's probably heavy.
It's a really good walker.
Yeah, really good at walking.
Maybe even like speed walking. I heard
Yeah.
that's a sport now.
They don't have to walk very fast, their legs are so long.
I guess speed is all relative.
That's true.
They're speedy.
This is galloping.
Yeah. What's that little bit? That little black bit?
Yeah, it's a parasite, probably a leech.
Ah, yeah.
Ooh.
We've seen that in a previous dive, haven't we?
Ali,
Let go of our friend.
Ali. Moet hulle get aan him.
I think it got tired.
See you later.
Hi.
There.
That was probably the most exciting thing that's ever happened to it.
It's going to be telling all of its friends, you're not going to believe this.
Mm-hmm.
You are muted. Never mind, muted. Okay. Thanks for waiting. Yeah, class of join them.
Yeah, as I was just saying previously, it's, now we are a training ship, and that's part of our staffing model here. We are a 24-hour operation, so we're always exploring when we're at sea. There's always something happen. And so we're staffed basically so that you have
Ethernet in that WCA bottle?
triplicates every single position on the ship. There's 3 people that have that same job, and that way we can cycle people in and out so people don't have to work more than a regular work day. And then within each triplicate, we typically have someone that is very senior, and on the other end of the spectrum, someone that's very junior. And we find that this is a great way of having some peer-to-peer mentorship, training, and not just training for and cross learning from senior to junior, but also the other way around. And it's
Totally.
so much we learn, especially for technologies are developing very rapidly these days. Yeah, if we can get a good zoom on the head. Yep, that's great. No, that was good.
Did we have an ID on that fish?
Oh, that's Eldrovandia, the halosaur.
Which is the best transformer name.
Mm-hmm. What did the transformers say when they transform?
Oh, help us out, viewers. I don't remember. I don't think it was okay, Roger. I think it was something else.
That was,
I know.
I was responding to Mike in the Data Lab, but that kind of fit.
You're doing great work. I would like that, like the giant robots showed down, and it's like, okay,
Time
Roger.
to transform. Okay, Roger.
We're gonna reboot this. It's gonna be great. I know we can trust we can count on our viewers at home. Thank you for the help. We got Autobots roll out and Autobots assemble.
We gotta assemble our Autobot.
Okay, Roger.
So for those folks who are following along with the dive plan, we just passed waypoint a while ago, so waypoint 2. So this track that we laid out for this dive, we started at about 3,000 meters on the northern end of this seamount, and the plan basically was we laid out a track a little over 2 kilometers going up the steepest portion of the seamounts toward the summit. So we hope to reach the seamount top at a depth of about 2,200 meters. Today's dive is going to be happening until midnight today, so midnight will be secured on deck. So still a lot of dive time, a lot of exploration time to go. Just as a time check, so we'll be leaving the bottom around 10:45 p.m. local time in Palau. So that's basically 9 hours from right now is when we'll have to leave the seafloor. We ascend at a rate of about 30 meters a minute. And for the plan looking forward, so we'll leave we'll have the ROV secured on deck here at midnight, and then we're going to be undertaking a transit, somewhat of a lengthier transit, a little over 13 hours to get us to our next ROV exploration site. That is one more seamount, and that's going to be due south of Angkor Island.
Oh, it's a black coral. Right? What is this? Oh, no, it's hydrozoan.
Very tricky.
Yeah, well, that's why we need zooms.
Yeah, I wouldn't feel comfortable.
I was like, that's super weird looking.
Megan, tell us what a hydrozoan is.
So hydrozoans are very small coral-like animals. They can come in many different forms, some that look very coral-like and some that look very jellyfish-like. So it's a very broad-ranging group of animals, and the ones that we saw, that one is called Hydroidalina. It's always attaching to other substrates like dead corals and sponges in particular, maybe other types of dead organic matter. So we see that encrusting on many different things, especially sponge stalks and coral skeletons.
Thanks, Megan.
Yeah, just reiterating, so yeah, we'll be pulling the ROVs off the seafloor about 9 hours from now, secured on deck at midnight, and then it's 13 hours transit to our next exploration site. That is yet another seamount that has never been mapped. It's quite a bit south of Angar Island. And so we'll have to map that site before diving it, so probably sometime tomorrow afternoon after around 2 o'clock or so, as we'll have enough mapping data acquired to be able to plan that dive. And that will be the second to last dive of this first leg of the expedition, Le Bou's Voyage 1. We'll have one more dive prior to heading into port on Friday, November 7th, a local date. I know it's a bit confusing for some of our scientists ashore that are following us from all over the world, and some of you are on the other side of the date line, so it's 1 day after your equivalent date.
Yeah, local time 8th.
And, and with that, I just also a big shout out to all of our scientists ashore who've been joining us all odd hours. Currently we have joining us Asako Matsumoto from the Chiba Institute of Technology in Japan, Dr. Christopher Kelly from the U.S. West Coast, formerly at the University of Hawaii at Manoa, Ikelau Oto, Chief Researcher at the Palau International Coral Reef Center, James Reimer, Professor at the University of Ryukyu in Japan, Jonathan Womack, Independent Researcher, Dr. Kenneth Sulak, retired from the U.S. Geological Survey, and Dr. Steve Askadich from the National Museum of Natural History, Smithsonian Institution in D.C. So thank you so much for joining us. Oh, can we get a look?
Shrimp is back.
Yeah.
Yeah, didn't you like the shrimp scoot? That was really fun.
That's great. Yeah.
The surface area of all those legs, like holding in the water column like that, it's really incredible.
Yeah, you can just sort of glide around.
It reminds me of, I don't know what they call them regionally everywhere, but we call them water skippers, the little bugs that glide on their
water
toe
striders.
tips. Yeah, water striders maybe. It's kind of like roly-polies. Turns out there's a regional name for all of those, but maybe water strider skippers are the same. Oh, so awesome. What a great view. Coming in for landing, legs tucked. So cool.
Good job, Shrimp.
Stuck the landing.
Perfect.
Super cool knowing the collaboration of so many scientists being here and helping out as well. Fun to have Nautilus Live viewers with us. If you're watching over on YouTube, feel free to come on over to nautiluslive.org and send in your questions. But we know we've got folks watching in the United States. It's getting late there. In Palau, midday for you all. In the UK and Germany, you're up late. Thanks for being here with us. In Canada and Australia and New Zealand and Japan. Really appreciate everybody tuning in and hanging out with us on the seafloor. So send in your questions for the team here. We'd love to answer what you're interested in learning more about, as we're all experiencing this seamount for the very first time. It's joining us on our exploration quest, coming right along.
It's just the journey of the shrimps right now.
Yes, indeed. All right, science team, May, as our geologist in the back row, maybe you can help with this one, but viewers want to know, how does the seafloor change over like geologic time? Maybe could you tell us, like, are we on some old seafloor here, some new seafloor? Like, where are we in the history in this part of the ocean?
Yeah, well, seafloor is really diverse across the ocean basins. So that question, how the seafloor changes over geologic time, really depends on where. In some areas, it can be changing really, really rapidly. And at active tectonic plate boundaries or at active volcanoes, new seafloor is being made. And at subduction zones, seafloor is being subducted under a continental plate. But the seafloor we're looking at here is quite old, and all of this sediment cover is also a good indicator that it's a very slow-going ecosystem, and much of what we're interested in on these dives is characterizing the life that has been able to grow in these sort of ancient environments. I really like Daniel's analogy on previous watches that some of these coral forests, these deep sea coral forests, are kind of like the ancient old growth forests of the sea. So yeah, it can be very old and very young, but what we're looking at here is quite old.
Yeah. We also know that sedimentation can increase across the ocean. It is not a one-to-one, like deep sediment doesn't automatically equal super old seafloor. It depends where it is, but when you're not out in a continental margin,
The pink.
you're adding sediment from marine snow, so that can be one indicator, but it's also possible to age rocks from the seafloor with geologic methods to understand how many millions of years old they are.
Yeah, definitely. And part of the geologic objectives of some of these dives is to collect nice basalt samples that are in place and not too weathered, so that we can do some, so that some scientists ashore who are collaborators can determine those ages of the lava flows and seamounts that we're looking at.
Oh, is it eating something?
It might be.
That's so cool.
It looks very relaxed. Just
6 arms, okay. Who are you?
I think this might be a Polasterius.
What gives you those clues?
It's kind of looks kind of like fingers inside a glove.
Ooh.
Texture. I don't know. Maybe that wasn't a great description.
Great.
But yeah, they kind of look a little more balloony.
Yeah, yeah.
They have a very unique texture.
I see that.
Jacob, if it's possible, I just wanted to see if we can line up. I want to try to get a good shot with the cinema cam. So why don't I just give that a shot.
And we usually see this one on rocks and not in sediment. But I feel like it's found like the one pebble in this whole sandy area. Yeah, and this star always has the 6 legs and tends to be pink.
Okay.
Yeah, so video, is it all right to zoom in, or
What's that?
is that okay to zoom in? Are we? Yeah, it's
Yeah,
okay.
on the cinema cam, yeah.
So interesting, kind of hovered over that rock like that. I didn't see its stomach out, but
Yeah, it could be eating something that it found. It does look like a feeding kind of posture, but maybe it's eating
Thanks,
something
Jake.
small. Oftentimes when we're in a coral garden, we'll see sea stars feeding on corals.
I almost got myself uninvited from Thanksgiving once by teaching my little cousins how sea stars eat. I was like, okay, now they take their whole stomach out of their mouth, and then you're going to wrap it around your plate, and then you're going to pull all the goo back up into your body. And everyone was like, not cool, Megan. Not really the time for marine biology facts.
It's always the time for marine biology
I
facts.
agree. I agree with you.
There's so many good facts, and I always like to share them at inappropriate times.
So science team, some viewers are writing in that they're observing a little more activity on this seamount versus yesterday's. If you were thinking about those two, how would you compare them? Any reasons why we might see more or less on one?
Well, it could be your location on the seamount. Well, first of all, every seamount is different. So it's like every house is different. Things are going to be in different places, right? But the location on a feature that you dive on is also going to affect what you're seeing. Sort of on the northern slopes of a lot of these seamounts tend to be more or the western slopes of these seamounts tend to be more sedimented, whereas the more windward exposed areas tend to have a little more scouring. That's not always true, but we do notice that kind of pattern. If we're diving on a ridge feature versus a steady slope, today we're on sort of a steady slope and not a exposed ridge feature. Those ridge features will have a current acceleration, so usually less sedimentation and more hard substrate in those spaces. And depending on hard versus soft substrate, you'll see a different array of animals. In these sedimented areas, we tend to see more shrimp and fish and echinoderms on the seafloor, whereas on the hard exposed substrate areas, we're going to see more corals and sponges and associated fauna with those types of animals. Depth is also a key factor here. The deeper you are, the different types of animals you're going to see from shallower areas.
Anybody have the depth range of yesterday's dive off top of mind?
Yeah, so yesterday's dive, we started around 2,400 and then made it all the way to 1,500 meters. So that seamount was a bit shallower than this one. The shape was also fundamentally different. Yesterday's dive seamount had this large flat top. Today's seamount has a summit that is about 2,200 meters, so 700 meters deeper than today's, and the shape is kind of more has a ridge type at the top, not a large plateau. In terms of the size of both features, these are big features. I think this is something that needs to, like, most people don't really wrap their head around. Is these are 10 kilometers or so in width, and that we hadn't mapped before yesterday, and so 10 kilometers in width, and then 3,000, 4,000, 5,000 meters tall from the bottom of the ocean. And up until yesterday, both of
Alright,
these
we can let
features
him
had
go.
not been mapped. And so we mapped them, and then just a few hours later, we used that mapping data to try to pick the track that we thought was most interesting. So we did a southern approach of the seamount yesterday. Today is a northern approach. So going across.
Is that another crevice?
Yeah, let's take a look at that. That's
It looks really similar to the other one.
yeah.
There's kind of that sediment kick out at the bottom.
And
Yeah.
there's a shrimp in
Like
there.
Sapiro.
That is
Maybe
funny.
the shrimp is doing this.
That would be very powerful for those shrimp.
Well, shrimp's got time.
How long does shrimp live?
I don't know. It depends on the shrimp. The
Really interesting.
deeper living animals tend to live longer lives.
Huh.
The shrimp has a really short rostrum.
It seems to be kind of the same length as the previous crevasse that we saw.
Very similar.
Yeah.
Well, now we know it's not just a coincidence. Something seems to be happening here.
Brian would like to slurp the shrimp.
Okay. Bridge nav. Can we hold position? Thank you.
Apparently they've seen this also on the previous watch, I guess. So this would be the third time they've seen these sediment crevasse.
Oh, interesting.
So all of our slurps are empty, so we can start with one.
All right.
Do you want more? Okay, cool.
Nope, I think it went in.
Oh, is there another shrimp at the bottom of the crevasse?
Oh, there is.
I just thought it,
Is that the same shrimp?
I don't.
it wasn't there before.
Yeah, okay.
I don't know if it escaped.
Now it sees an empty crevasse and it's like, okay, this is mine now.
Or did it jump out?
It might have jumped
as we
out.
were slurping.
Yeah, there's nothing in the jar yet.
Yeah, it might be that little one.
It's in the crevasse. It's like down in there.
If you're watching on channel 3,
Nah,
you
it's
can
still in
see the
there.
bucket cam, which is what we're looking at.
Yeah, it's still in the crevice.
I mean, this is obviously an effective protective habitat. In all these moments, I feel like I lean forward towards the screen. I'm going to be able to see better down into the...
Yeah, I don't know. I don't know if we'll be able to get it. It's still in there. Oh, it's coming out.
Oh.
Oh, it's up alongside on the outside.
It's a very smart shrimp.
I feel like it's messing with you.
Oh, there we go. In the end.
Let's see what it does.
Oh, it's
Oh,
in
there
there.
it is. It went in.
Okay. It was smaller than I thought it was going to be.
Some folks watching on channel 3 seeing in that bucket cam some of those larger sediment chunks. What are those shells, or what might those be?
Yep.
Yeah, those could be shells or little bits of carbonate.
Or just kind of globs of sediment
Yeah,
that have...
conglomerate. We have been seeing little shells, though, in some of our slurps.
Gotcha. Yeah, if you're watching on channel 3, you've got that top-down view of Jake and Sarah there. You can see the manipulator arm in use with the mini controller. I'll go back, Daniel. So looking at the dive plan, this this feature is very big. 3,000 meters tall, is that right?
Yeah, I mean, it depends where you measure, but yeah, they typically start at 5,500 meters is kind of the bottom of the seafloor there when you're not in the trench. And yeah, this being about 2,200 meters, so 3,000 meters in elevation thereabouts, and then probably at least 3 times as wide as it is tall. And yeah, it's very interesting when you have, especially what we're going to be doing over these next 3 weeks is diving Basically, we started up in the north, did a few dives north of Alaska Reef, and then we're going to be working in a clockwise fashion around the main island group.
Acanthonus armanis.
Yeah, I think just for scale for a second, I love measuring in strange things. You know, so we're talking about the scale of that mountain. That is 12 of the tallest points in Palau stacked on top of each other to be the size of this feature.
What's the tallest point in Palau?
It is about 250 meters, and maybe Mike can help us pronounce it.
So it is called Ngar Alus, and it is located in Ngartmao State, which is on the western coast of Palau, and that's the highest point, yeah.
Thank you so much.
Yeah.
Ngar Alus?
Yes.
Awesome. I'm working on it, slowly but surely. If you haven't been checking out the Palauan word of the day, follow us at Nautilus Live on Instagram.
So what was the word of the day today?
We haven't posted today's word yet. I believe it's going to be horizon. It's going to be the word. I had beautiful clear skies to really enjoy that sky-ocean boundary.
Yeah.
We could ask Ilani when she comes back. She's doing the word of the day today.
Yeah, so as I was alluding to, over the next 3 weeks, we're going to be diving in a clockwise fashion, targeting various different seamounts. So started in Velasco Reef, so did a couple dives up there in the north. Then we were just on the eastern corner yesterday, and then we're now starting to move southeast and continuing on. Headed to port on Friday, and then the second part of the expedition is going to be working in the southwest islands and around there, and then ending up in the western coast. And by doing that, basically we are targeting seamounts. They're all exposed to very different current regimes. So here in Palau, the main currents are basically coming either from the west or the east. So you have the north equatorial current, the north equatorial counter current, and they basically then intersect with the Palauan main island group, and then on the downside stream, so west of the main island group, you have all these eddies, turbulent features that come off. And so there is a predominant current flow, and so these seamounts are all going to be exposed to very different oceanographic conditions. So we're targeting things that have lived in a different environment. We're also trying to target seamounts all across their depth range. So we're going to be, the deepest features we're going to be targeting are 4,000 meters deep, and then we'll do a dive that's going to be as shallow as 500 meters or shallower probably. So really trying to tease apart all the different environmental factors that create ecosystems that are of high conservation value. And seamounts in general are known, they're known in layman's term, people typically refer to them as the kind of the rainforests of the deep. They're hotspots of biodiversity in an otherwise somewhat less populated deep ocean. There's over 30,000 seamounts globally. Very few of those have been studied. Here in Palau, over 200 of them, and only a handful of those have been studied, and we'll, over the course of these next 2 expeditions, do a lot, increase significantly what we've known. Oh, this is very interesting.
Yeah.
Huh. Yeah. Very different terrain.
Giving some kind of moon texture.
Wow.
Kind of like mudstony.
Yeah.
Yeah.
But no indication that it like slid down, or at least not recently.
Looks like maybe
tracks
ahead. There's like a.
Oh yeah. Shelf.
Yeah, mini shelf.
Just a little bit of a slump.
Oh yeah, look at that. Bubble.
Interesting. Oh yeah, you can almost see the section like it was broken off.
How many years of marine snow?
Yeah.
Ooh, good question. Does anybody know that? How quickly marine snow falls here?
I'm not sure the sedimentation rate here. Do you know, Daniel?
I don't know, but I know that there's a team from Taiwan that did an expedition last July. And it's a little bit more of a regular thing, I believe. It's a multi-year project that are going to be coming out here and laying sediment traps to try to quantify that, try to understand. So yeah, the deep ocean is obviously important for many different reasons. It's the largest ecosystem on Earth. It's also the largest carbon sink on Earth. So understanding how greenhouse gases that we emit into the atmosphere and then eventually dissolve into the ocean and then eventually get buried into the deep ocean. Understanding that carbon cycle that happens over very long time scales is really important for understanding, climate change and how the Earth is going to be coping with these things. And
Can we get a zoom on some of these layers?
yeah,
Sure.
How hard do you think
You
it
wanna push in?
is? Do you think it'll crumble really easily?
I
or-
think it would
Yeah,
crumble
I think so
pretty
too.
easily, yeah.
Yeah, that looks very.
It's,
I think we saw some evidence of the crumbliness a little earlier.
Just below, yeah.
yeah, this is not very consolidated. What are the contour lines in HIPAC? Like, what kind of slope are we on?
So the lines are 10 meters apart in elevation.
Nice.
There's so many different kind of grain sizes and colors in it. It's really interesting.
Yeah. Some kind of landslide.
Yeah, with the shrimp.
Oh, another shrimp. Very cool.
Yeah.
I don't think we've seen this kind of slump before.
Perfect time for this viewer question. Someone interested in the in fauna, the creatures that live inside the sediment and the mud. Has there been any, I don't know, I guess gravity core, drop coring that would look at creatures living in the sediment here?
Yeah.
Did you ask if there was any sediment coring?
Or question is about little creatures that live in the sediment, thinking about worms and things that burrow in.
Well, we did just collect this shrimp-like creature, and I did want to make a note that one of our scientists ashore, Kenneth Sulak, mentioned that it might actually be a blind lobster, which are known to excavate deep burrows into soft substrate. So that would be one example of a critter living inside of the sediment. But yeah, in terms of worms, we have collected some, and we have been seeing these kind of crop circle-like sediment formations on previous dives, which we believe are from sediment burrowing worms.
Push in there?
It's coconut?
Yeah, I was wondering if it was.
Looks like it, yeah. More debris.
Oh, so interesting.
Did we want to get sediment cores on this dive?
I don't think so. I think we're good on that, right, Daniel? Or
Yeah, I mean,
It definitely is. Yeah.
I think the request was more around manganese, like.
it was a wide request, yeah, so
Yeah.
we could. This is probably one of the better places to get it, but
We can do it if you think that.
yeah, it was just a request for the 10 top centimeters, so.
Well, we know that
soft.
at least there is 10 centimeters here.
Yeah, exactly.
Jake, are those clouds from us?
Probably,
Cool.
but I don't know.
Here's a fish.
Folks are always interested
Straight
if there's
ahead.
stuff stirring up around us or with us. Hello, fish.
Push in on the fish.
All right, I'm going to give the watch back to Made. Have fun.
This is a cusk eel called Bazazetus.
All right, come on.
So yeah, for those who are following along in the dive plan, it looks like we're almost in the middle between waypoint 2 and waypoint 3. So that would put us in about 800 meters from where we started when we reached the seafloor several hours ago. We've also climbed about 300 meters of vertical relief, so we're at currently at 2,600 meters. We'll continue moving our way up the slope, documenting what we see. We'll also be taking a Niskin bottle and about 100 meters further up at 2,500 meters. Just a shout out to our folks here on board, our scientists from the Palau International Coral Reef Center who have been working extra long hours in the wet lab, processing the environmental DNA samples, collecting not just at the surface but also at depth, and then taking several hours after each dive and after each sample collection. And so yeah, maybe Maikani, if I can put you on the spot, maybe if you can talk a little bit about that program and the importance for monitoring the sanctuary.
Yeah, thanks for that, Danielle. So the Pickerick eDNA monitoring
There's
program,
another fish.
it started around 2021, where we have a bunch of different sites within PNMS, also inside the artisanal and domestic fishing zone. So we have a bunch of sites in those areas where we collect water samples, we bring them back, and we filter them. And within those samples
Push in there on the
are little DNAs of fish and other marine organisms that we use to determine which kind of marine organisms are living within PNMS or within Palau's waters in general. So we've done about 4 collections since 2021, and we're in the process of collecting more water or doing more field work for this project. But yeah, it takes a while to filter them, about 2 to 3 hours, maybe 4 if the water is really murky. So Pierre and I have been working extra long hours just to filter these water. But yeah.
Was it a bit faster with the double pump?
Yeah, so it's been going a little faster. I think the samples we had yesterday and today were just a little murky.
Okay.
So it was going a little slower, but the other pump is really, really fast. So Pierre did it yesterday, finished in like 3 hours.
Nice.
So yeah.
A question on that, Maikani. So how exactly do you distract the DNA from the water samples?
So we would have